We now continue the previous lecture, the lecture about stress and memory, and we finished that lecture with a very disturbing, provocative question. You'll recall in the final minutes of that lecture, what we focused on was the consequences of major long-term stress for the brain, for the memory systems in the brain, and we saw lots of bad news. Chronic stress and the declarative memory that the hippocampus specializes in disrupted. Chronic stress, less oxygen, less glucose delivery to the brain. You begin to disrupt long-term potentiation, that increase in excitability. You begin to cause atrophy of neurons in the hippocampus. You suppress neurogenesis, that wonderful new phenomenon. You make it harder for neurons to survive all sorts of insults. And at an extreme, you can actually kill neurons. So the question we were left with there is, what about humans? How much of this applies to the human brain? And much of this is impossible to study in humans, not like you could study in laboratory animals, as described before, because you can't tell if individual neurons are atrophying in the human brain. You can't detect whether there is long-term potentiation in particular connections between neurons. But one thing you can do that's quite pertinent is to take advantage of brain imaging techniques, ones where you put somebody in a scanner and you can see the size of different structures of the brain, the level of activity, CAT scans, MRIs, things of that sort. And what you can do with those approaches is look at the size of the hippocampus. And what those studies have provided are the starts of some pretty disturbing conclusions. What we know by now is what's going on. An awful lot of those sources, chronic stress causing atrophy, neuronal damage, all this stuff in the lab animals, is a function of the excessive exposure to glucocorticoids. So we start off with the first domain of humans where there is excessive exposure to glucocorticoids. Back to the immunology lecture, that whole business about what glucocorticoids can do is suppress immunity, and that business that when you have an immune system that spirals out of control into autoimmunity, glucocorticoids are good for coasting you back again. And as a result, there are hundreds of thousands of people out there taking high-dose glucocorticoids, synthetic versions, long-term to control their lupus, another autoimmune disease, their rheumatoid arthritis, whatever, and suddenly you come up with the inevitable question, do folks like these begin to have the sort of memory problems that are associated with hippocampal dysfunction? Now, these are extremely tough studies to do because by definition, if you are marinating somebody in synthetic glucocorticoids, it's because they've got a serious disease. And if there's cognitive problems, is it due to the disease? Is it due to the steroids? And it has taken some very subtle, difficult studies, case control studies, where you match two people for age, education, gender, all of that, and the severity of the disease, and you take ones where it's been treated over the years with non-glucocorticoid, non-steroidal anti-inflammatory drugs versus steroidal glucocorticoid ones, and then look at memory, and just the first hints of a literature showing the more glucocorticoid exposure, the more problems you have with hippocampal-type memory. So that's not good. Moving to the next version that of relevance to humans, a disease, in fact a syndrome of diseases, called Cushing's disease, Cushing's syndrome. Any of a number of different types of tumors where the commonality is you secrete boatloads of glucocorticoids. And you've got all of the side effects that are associated by now, diabetes, hypertension, all that sort of thing. And inevitably one then has to ask, what's going on up in the brain? Do you get the sorts of memory problems associated with a hippocampus that is not doing its best? Yes, indeed. It's often called Cushingoid dementia. And what about the hippocampus in terms of structure? First brain imaging studies some years ago showing that long-term chronic people with Cushing's disease, typically people have Cushing's for about 2 years before it's diagnosed, major long-term Cushingoid bathing in glucocorticoids, and you get atrophy of the hippocampus. The more prolonged, the more severe the glucocorticoid excess, the more hippocampal atrophy. Now, is this due to dead neurons? Almost certainly not. because equally interesting studies have shown if you then go in and surgically correct the tumor, get rid of the excess glucocorticoids, over the next year or so, the hippocampus comes back to normal volume. What's this about? This is almost certainly that atrophy of individual neurons, because what the laboratory studies show is with the abatement of stress, with the abatement of glucocorticoid exposure, the processes grow back. So the hippocampus is able to shrink and seemingly some capacity to recover, and the memory processes come back to normal. Now, the next human version is truly profoundly troubling, which is an emerging literature, well-replicated studies showing that chronic major league depression can be associated with hippocampal atrophy. As already alluded to, and as what will fill up 2 different lectures, depression has much to do with stress. Depression is intertwined in many cases with glucocorticoid excess, and what these studies have shown is atrophy of the hippocampus. The more prolonged the depression history, the more atrophy, the more memory problems. Is this reversible? To date, the majority of studies have shown that it's not. With the abatement of the depression, when it's gotten under control with the correct antidepressants, when the depression goes away, you don't necessarily get full recovery of hippocampal volume. Caveat, fine print here, have a depression, have a depressive period. Do not get into a fraught state here of worry. No evidence yet that everyday clinical depression can cause this. This is horrendous, massive, serious depression associated with this. Next version that might be relevant, a human disease, a disease of people with horrible exposure to stress, some traumatic incident producing PTSD, post-traumatic stress disorder. And what is seen there in a large number of studies from recent years is, once again, atrophy, loss of volume of the hippocampus, preferentially the hippocampus, and the more severe the traumatic history, the more atrophy, the more memory problems. Now, this happens to be a wildly controversial, incendiary subject built around this confusing issue: depression, very often elevated glucocorticoid levels. Cushing's disease, by definition, elevated glucocorticoid levels. Give somebody synthetic glucocorticoids and you raise their glucocorticoid levels. It remains very controversial whether PTSD is associated with elevated glucocorticoid levels or remarkably lower than normal levels. And mixed results in the literature. It's not entirely clear what's going on. Nonetheless, in the cases where it's below normal levels, what you see is enhanced sensitivity to the glucocorticoids. So the PTSD story is quite confused at this point, but what is far clearer is the evidence that lots of synthetic glucocorticoids, excess glucocorticoids in Cushing's disease, excess glucocorticoids in the severe cases of depression, you get loss of volume of the hippocampus. The more severe the insult, the more volume loss, and the more problems with the sort of memory that the hippocampus specializes in. Okay, disturbing bad news, but this is disturbing bad news at some of the extremes of things that can go wrong. Tumors, major depression, all of that. What about more everyday stressors? What about, for example, chronic occupational stressors? And there was a remarkable, fascinating study that was done some years ago, published in the journal Nature, which is probably the most prestigious journal in the world. And what this was about was looking at people with a certain occupational stress. These were flight attendants for 2 different airlines, both of them that do intercontinental flights, and the 2 airlines happen to differ after you do an intercontinental flight with a massive switch of circadian cycle-inducing dramatic jet lag. How many days do you get off from intercontinental flights in order to recover? And there was the nice airline where you had 15 days to recover. And then there was the mean stepmother airline where you had 5 days to recover. And what the study showed, and it was perfectly done, controlling for the total number of time shifts that somebody had done professionally as a flight attendant. So it's not that one group had more of these shifts. The two groups merely differed in recovery time. And what was shown was those folks working for the airline that gave you only the 5 days to recover, smaller hippocampus and more memory problems. What I thought was very interesting, actually two things. One was that the paper carefully went about not mentioning the names of these airlines, because I assume if they had mentioned the 5-day one, they would have gotten their Keister's suit off for this. The other was, it struck me somewhere in there, that also undergoing all of those time shift stressors are the pilots. And they were unmentioned in the study, but it seems kind of disturbing. I mean, it's bad enough to get the memory problems where you can't remember if the person wants ice cubes in the milk or not. But if you're getting problems like you can't remember if this little old switch goes up or down in the cockpit there, this is not a good thing. Nonetheless, first evidence that occupational stress can have some of the same consequences. Finally, the realm of normative aging. And this is wonderful work done by a neuroscientist, Sonia Lupien, at the University of Montreal. And what she has done is look at what glucocorticoids might have to do with successful aging. A subject that will come in an entire lecture, but just briefly foreshadowing here, in this study of hers, she took 70-year-old individuals and characterized them at that point, the size of their hippocampus, brain imaging, their glucocorticoid levels, and then came back 5 years later. And what she saw was, for those people whose glucocorticoid levels have risen over those 5 years, they tended to lose hippocampal volume and tended to get more memory problems. So at this point, this is a very preliminary literature. Nonetheless, it suggests all the bad news of the last lecture in terms of what chronic stress, chronic glucocorticoids can do to memory processes might very well apply to our own brains. Now we transition to the other topic in this lecture, which is the fact that your brain is not just good for playing the piano and going down flights of stairs, and your brain is not just good for learning and memory, your brain is also good for judgment, for impulse control, for executive decisions, and all of this involves a part of the brain, briefly mentioned, a part of the brain that is so interesting called the frontal cortex. The frontal cortex is absolutely fascinating. Cortex, the surface of the brain, frontal cortex, the surface in the front of the brain. The frontal cortex is such an interesting part of the brain. I have spent the last 25 years of my life studying the hippocampus. The hippocampus has been good to me. I love the hippocampus, but I've become badly infatuated with the frontal cortex in recent years because it does such interesting stuff. Yes, learning and memory is cool. The frontal cortex, though, is amazing. The frontal cortex is arguably the most human part of the brain. Proportionately, we've got more complex frontal cortices than any other species out there. It's the most recently evolved part of the brain. Frontal cortex, a big-time frontal cortex, is virtually a primate invention. We're humans really running with it. What does the frontal cortex do? All sorts of complicated things, regulating executive behaviors and strategizing on the very simplest level. What does the frontal cortex do? It makes you do the harder thing when it's the right thing to do. It makes you do something that's difficult when that's what you should be doing. The frontal cortex, okay, you want some terrible, utterly inapplicable metaphor. Frontal cortex is the nearest thing we've got to a superego, the nearest thing to a part of the brain saying, I wouldn't do that if I were you. You were going to regret it. The frontal cortex has this role manifest in all sorts of domains. For example, the frontal cortex makes you at a party do the right thing even though it's harder. You've just been served the most horrendous meal you can ever imagine, and it's your frontal cortex that kicks in that says, whoa, this is delicious, I've got to have the recipe. The frontal cortex is what gets you to compliment somebody's hideous new hairdo. The frontal cortex keeps you from being a serial murderer. It keeps you constrained. It keeps you in line. The The frontal cortex is also something that kicks in a very dramatic way during early development. Most of us way back when had to suddenly get a lot of frontal function when we learned to be toilet trained. And that is something that soon moved from frontal executive control to implicit pathways. Most of us don't have to think consciously anymore about being toilet trained, but early on, that was one of the big initial frontal tasks that we had to master. Okay, so frontal cortex. Frontal cortex, all sorts of fascinating other features of it. And as just became clear, part of development, child development, is your frontal cortex maturing. And you can show the fact that kids don't have a whole lot in the way of a frontal cortex by brain imaging, things of that sort, and you find out something absolutely astonishing. The frontal cortex is the last part of the brain to fully develop. Your frontal cortex is not fully developed until around age 25. That is astonishing. That explains an awful lot of behavior in freshman dorms. That explains all sorts of things. The frontal cortex takes forever to develop, and thus childhood is filled with a frontal cortex that doesn't work very well yet. You can show this wonderful study, this wonderful experimental paradigm, where you take kids, and what you do is you put the child in a room, and you put a marshmallow in front of them, and you say, okay, here's the deal. I'm going out of the room. I'm going to come back in a while, and if you want, while I'm out of the room, you can have that marshmallow. On the other hand, if you don't eat it, when I come back, you can have 2 marshmallows. In other words, can you regulate your behavior, regulate this impulsive desire to grab that marshmallow? And kids very early on are awfully challenged in order to hold on and not grab the marshmallow early on. And in fact, there's a literature by now showing you take like 4-year-olds, and the longer they can hold on not taking the marshmallow, the more frontal regulation they have of their behavior. And that is in part predictive of things like SAT scores a decade later in high school. This is the mark of a frontal cortex that is developing in a very impressive manner. Although, as a parent, what I've discovered is there's a much easier way of measuring the degree of frontal development, which is play hide and seek with a child. Here's what happens. You're playing hide and seek, and the kid is hiding, and you count up to 10, and you come into the room, and you say, ready or not, here I come. Ooh, where are you? And suddenly the kid says, I'm right here under the piano, because they can't stop themselves from saying where they are because they got like no frontal cortex. Or you now switch, and now you're the one who hides, and they're over there counting up to 10. You go 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, because they've just learned all these numbers, and all these numbers are so exciting, and they can't stop themselves at number 10. This is exactly what a brain without a whole lot of frontal function looks like. There's another realm in which you can get brains without a whole lot of frontal function, brains that have been damaged. A remarkable finding, which is a substantial percentage of people on death row in this country have a history of major concussive head trauma to the front of the brain. And this will come back to haunt us in a few minutes. The implications of that. The other version of a frontal cortex that's not working very well is when you get a wildly violent criminal act by someone who is young enough that their frontal cortex is not developed all that much yet. And the possibility of this is what motivated a Supreme Court decision a number of years ago saying that you cannot execute somebody for a crime they did under age 18. And the decision explicitly talked about how some parts of the brain were not fully developed at that point, that there was some degree of impairment of frontal regulation of behavior. Now, as I mentioned, humans have the biggest, most dramatic frontal cortices out there, recently evolved. Other species have frontal cortexes too. Rodents have them. Primates have them. They don't have a whole lot. Take, for example, monkeys like baboons. And baboons are schemy, competitive, backstabbing animals, but they're not very strategic about it because at all sorts of critical points, they blow it because they're wildly impulsive animals. For example, baboons can form cooperative coalitions, two males who help each other out in a fight. And what you wind up seeing is this should be a brilliant strategy because four hands are better than two, or two tails are better than one, or whatever. And a stable cooperative coalition should be unstoppable. But with an astonishing frequency, the fight finally breaks out, and the guy spins around and attacks his coalitional partner. Because how could he resist? Because he's only got like 7.5 frontal neurons. And I remember some years ago sitting at a primatology conference looking at a film about primates that have really good frontal function, apes like chimps and such, and we were watching a film that was called Chimpanzee Politics, looking at chimpanzee political power-grabbing strategizing, which they're really good at, and I was sitting next to another baboonologist, and at some point we turned to each other after some amazing chess move that one of these chimps had just pulled off. We turned to each other and said, "My God, that's what a baboon would love to be like if it had a shred of self-discipline or gratification postponement." And they don't, because they don't have much in the way of a frontal cortex. Transition to apes, chimps, for example, and they've got a lot more frontal cortex, a lot better regulation of impulsivity, but they're still not in the range of a human. And you can show this with a study that looks something like as follows. Here you have, in this hand, you've got 1 M&M. In this hand, you have 5 M&Ms. The chimp has a choice as to which hand to reach for. The rule is, if it reaches for the 1 M&M, you quickly pull your hand away, and the chimp gets a reward of 5 M&Ms. If the chimp reaches for the 5 M&Ms, you quickly pull your hand away, and the chimp gets a reward of 1. In other words, if the chimp is disciplined and ignores the 5 M&Ms right there, and instead is going for the tougher decision, going for the 1 M&M, it gets the much bigger reward. Chimps cannot do that. They are ready to reach for the 1 M&M to do the more disciplined thing, and they just can't stop themselves. They reach over for the 5 because the chocolate is right there in their face and it smells so good, and they don't have enough frontal neurons to make them do the right strategic thing. Go for the 1 M&M because you get the bigger reward. But now you make a very important elaboration in the experiment. Now this hand has 1 little chip of wood. This hand has 5 chips of wood. Reach for the 5 chips, pull the hand away, and the chimp gets 1 M&M. Reach for the 1 chip, pull the hand away, and the chimp gets 5 M&Ms. The same sort of deal if you were more disciplined and reach for the 1 piece of wood, you get a bigger reward. And now chimps can do this before the stimulus, because the stimulus is less salient, it's less in their face. The chocolate, I can't stop myself. Wood chips, the chimp has enough frontal function to go for the single piece of wood and thus get the bigger reward. Now, as alluded to when talking about people who have come into the criminal justice system with a history of concussive head trauma in the front, history of frontal damage, there is a whole challenging world out there of what happens when people get frontal damage. And in terms of organic impairment, a problem with the brain that the courts generally recognize, there is the gold standard out there, which is when somebody is so organically impaired, so neuropsychiatrically troubled that, as the standard goes, they can't tell the difference between right or wrong. Trivia fans, this is something called the McNaughton rule in criminality, and it often goes with people with severe schizophrenia where they can't tell the difference between right and wrong. Get somebody with frontal damage and they can tell the difference between right and wrong. They simply can't regulate their behavior. They don't have volitional control. And these are often people, in fact, who can verbalize what the appropriate rules are, can verbalize what the strategy should be, could verbalize saying, I know I should reach for the 1 M&M because I get a bigger reward that way, and then they go for the 5. because they can't control their behavior. And this is a huge challenge in the criminal justice system. This is not McNaughton, you can't tell the difference between right and wrong. This is you can tell the difference, but because of an organic impairment, you can't regulate your behavior. And in my view, there's way too few states out there that recognize this is as much of an impairment as the right and wrong dichotomy. Okay, so now we've got a pretty good sense about how the frontal cortex works, how it evolved, how it develops during growth, during childhood, what happens when it's damaged. So what does stress do to frontal function? What does stress do to the frontal cortex? And what you wind up seeing is lots. And once again, the usual suspect, it's mediated by glucocorticoids. Back to our endless theme here, hormones, brain chemical messengers, etc. They work by way of binding to their specialized receptor. Thus, glucocorticoids work by way of glucocorticoid receptors. You look in the brain, and there's two areas that have glucocorticoid receptors at far higher levels than elsewhere in rodents and non-human primates and humans. What are the two areas? You guessed it from last lecture, the hippocampus, and from this lecture, the frontal cortex. These are the two hot spots in the brain when there are sufficient amounts of stress and glucocorticoid excess that you're beginning to mess with how the brain works. It's the hippocampus and frontal cortex that go first. Now, the last lecture, dribbling over into this one, really looked in great detail as to what happens in the hippocampus. With chronic stress, chronic glucocorticoids, that's that whole picture of atrophy, potential recovery, dead neurons, no more neurogenesis, that kind of thing. What's going on in the frontal cortex with chronic stress and glucocorticoid excess? Far less studied, a very new literature that's just emerging, but what you tend to see is atrophy of neurons in the frontal cortex. Atrophy in terms of processes retracting, same deal as in the hippocampus. The frontal cortex, again, the surface of the brain, and it comes with a number of layers, tends to thin out. You've got less complex connectiveness there, which thus is not surprising if you're having neurons disconnecting. Very similar picture to in the hippocampus. What's an extremely challenging finding at this point is what happens after the stress is over with. What we heard in the hippocampus relevant to Cushing's disease is after you've got the atrophy of the hippocampal neurons, they can regrow, they can reform their connections, and things go back to normal. What's known at this point, very, very recent studies showing in the frontal cortex, yes, stress glucocorticoids, you get atrophy of these neurons, and after it's over with, yes, they grow back, but they grow back with different sorts of connections. What does that mean? Nobody knows at this point. All that's known is lots of stress, lots of glucocorticoids, and the frontal cortex thins out, atrophies, almost certainly isn't working as well, and in the aftermath of it, it's going to work differently than it did before without anybody having a good sense yet of what differently means. Now, many lectures, oh so many ago, we were looking at the consequences of sustained stress during child development, everything about growth, prenatal stress, etc. Back to that point about the frontal cortex being the last part of the brain to mature, what happens with stress during childhood? What does that do to the frontal cortex? And there is such a disturbing, such a depressing literature emerging out there having to do with childhood development of the frontal cortex and socioeconomic status. Your degree of poverty, education, the wealth of the neighborhood you live in, that sort of thing, socioeconomic status. And a literature has been emerging for some years suggesting by some appallingly young stage, by like kindergarten, there are already differences in socioeconomic status in terms of how well kids have their frontal cortex working, how well they do emotional regulation, gratification, postponement. And the first studies are coming out now showing that those correlate with differences in metabolic rates in the frontal cortex. Mind you, the usual caveats, this is a statistical phenomenon. This is on the average what you see. There's huge amounts of variability. Nonetheless, a totally disturbing suggestion that by an extremely young age, your socioeconomic status, the sheer, utter, damn blind luck you've had in terms of which household you got yourself born into is already having an impact on your developing brain, how readily you control your impulses, how readily you control your behavior. This is a theme that will come back in a whole lecture later on, having to do with stress, health, and socioeconomic status. Now, lurking in here is a very important subtle point, which is there's huge excitement these days about what genes have to do with behavior, that sort of thing, sequencing the human genome. Keep this in mind. Interesting implication. If the frontal cortex is the last part of the brain to fully mature, by definition, the frontal cortex is the part of the brain least constrained by genes and most shaped by environment. So what we've seen here now are all sorts of circumstances with chronic stress, chronic glucocorticoids, where the frontal cortex doesn't work as well. What is it that we've explained here? This too familiar circumstance where during a period of stress, you make some decision that is just asinine, which at the time seems utterly brilliant and insightful, and you spend an awful long time afterward dealing with the adverse consequences. You really do need to have a nice functional frontal cortex, and chronic stress doesn't do good things to it.